Hi, I'm Caitlin Simopoulos, and I'm a postdoctoral associate at the University of Ottawa in Ottawa, Ontario, Canada. Today, I'm going to be talking about how MetaProClust-MS1 can cluster metaproteomes using MS1 profiling only, which is our approach for more efficient, resource-conscious, and intentional metaproteomic research. First, I'd like to introduce the human gut microbiome, which will be the system used in our example datasets presented near the end of this talk. The gut microbiome is the collection of bacteria, fungi, archaea, and viruses that live in our digestive tracts, as well as the entirety of their genetic information. The gut microbiome plays essential roles in our lives, such as by fermenting dietary fibers or providing protection from invading pathogens. As such, the function and dysfunction of the gut microbiome can influence our health and well being. And many diseases and conditions have an association with the gut microbiome, such as immune associated diseases like inflammatory bowel disease and asthma, metabolic disorders like diabetes and obesity, cardiovascular diseases, and even our mental health can be influenced by our gut microbiome. And while our gut microbiomes can be influenced by our genetics, our lifestyles and environments have the largest influence. For example, people living in different geographical locations display different gut microbiota signatures. Gut microbiomes of infants differ whether they were delivered vaginally or by C-section, and our microbiomes change as we age, and they'll look different in newborns compared to older adults. Even stress and exercise levels can affect our gut microbes. But finally, the current focus of the lab of Dr. Daniel Figues is how xenobiotics, such as medications or drugs, affect the gut microbiome. In particular, we're interested in understanding how different drugs or compounds may affect the microbiome by observing the taxonomic and functional changes of these microbes under different conditions, specifically when gut microbiomes are cultured with drugs. There's evidence to suggest that many drugs have impacts on the gut microbiota, and some drugs are even activated or inactivated by our gut microbes. But we're still working on characterizing the effects of all drugs on a wide range of microbiomes. In particular, it's essential to characterize the robust effects of drugs on gut microbiomes, and this requires large-scale and high-throughput experiments. Metaproteomics, the study of proteins from multiple species, can be used to profile both the taxonomic and functional compositions of microbiomes. And our lab recently published RAPIDAME, an assay for large-scale studies of the microbiome. As proof of concept and as an example of large-scale gut microbiome studies, the lead author of this study, Dr. Luoyan Li, looked at the effects of 43 different drugs on 5 microbiomes. This is a total of 215 different samples, and this doesn't even consider replicates. In metaproteomics, we most often use data-dependent acquisition by tandem mass spectrometry. This is where we first take our extracted peptides and separate the peptides by liquid chromatography. From there, a first mass spec measures the intensity, retention time, and mass to charge ratio of a molecule. And molecules with the highest intensity are chosen for further fragmentation. And retention time and mass to charge features are then used for peptide identification that were identified in a second mass spec or by a second mass spec. A challenge with tandem mass spectrometry is that it's not time efficient in high throughput or large scale metaproteomic studies. In fact, tandem mass spectrometry data acquisition can take a really long time, and it can take upwards of 260 minutes for each sample if we're looking at deep metaproteomics. So if we think back to a couple slides ago when I mentioned an experiment that used 5 different microbiomes treated with 43 different drugs, this could take over 38 days just to acquire the data, and this isn't even considering experimental time or data analysis time. In addition, data-dependent acquisition is limited to high abundance molecules that are measured by the first mass spec, as I mentioned in the previous slide. So this leads us to question whether we can use the information acquired at MS1 to complete a metaproteomic screen. So to test this, we have 2 research questions. Number 1, can we identify robust shifts in gut microbiomes at the MS1 only level? And if we can, are these shifts the same that we identify using a traditional or more classic tandem mass spectrometry approach? To answer these questions, we introduce MetaProClust-MS1, a computational framework for screening metaproteomes. Briefly, this framework starts with quantified features by MS1, and then ICA matrix decomposition is used. This is followed by k-medoid clustering of MS1 features, and then cluster eigenfeatures are calculated and represent the feature intensity of each cluster. Finally, the correlation of treatment with eigenfeature is calculated and is used as a distant matrix for the final treatment clustering. In more detail, we first detect and align MS1 features. This can be done using a variety of different software, but we chose to use OpenMS for its flexibility and command line interface. Once our features are quantified, a robust independent component analysis, or ICA, implementation is completed using the number of components that are automatically chosen by principal component analysis. ICA is used because it's a type of blind source separation, where it separates mixed signals into individual sources. Once decomposed, the S matrix will represent the features of the dataset and their projections or contributions onto the components, where components represent latent variables, potentially representing a molecular function or process. Once the matrix is decomposed, features are clustered by k-medoids clustering. Eigenfeatures are calculated from the intensity values of each cluster and are used to represent the intensity of a cluster as a single vector. Eigenfeatures are then correlated with a binary model matrix of sample treatments. We tested MetaProClust-MS1, or MPC, on real microbiome data, where we treated a gut microbiome with 5 different drugs already known to have effects on the gut microbiota at 3 different concentrations: high, medium, and low. Drugs included in our analysis are an immunosuppressant, an antibiotic, an NSAID, an H2 receptor antagonist, and Tylenol. We acquired data at 2 different levels. First, at using tandem mass spectrometry and a 60-minute gradient, and then is again using at only the MS1 level, using a short 15-minute gradient. First, I'd like to present the tandem mass spectrometry results from MPC clustering. On the left is a PCA biplot that demonstrates the discriminative ability of tandem mass spectrometry. The right heat map and accompanying dendrogram may be more interesting. The rows of the heat map describe the correlation coefficients of each peptide cluster to each drug treatment. This correlation information was used for the final hierarchical clustering of treatments, represented by the dendrogram up top. The stars represent robust clusters as identified by bootstrap analysis. In our results, we see 3 main clusters. The cluster on the left contains all concentrations of ciprofloxacin, an antibiotic, as well as the high concentration of diclofenac, an NSAID. This could potentially indicate that high concentrations of this NSAID act like an antibiotic. The MS1 only results show a similar story. Again, the PCA shows that MS1 only data can still be discriminative in terms of drug treatment. And the MPC clustering shows fewer robust clusters as identified by bootstrapping. However, we again see that a high concentration of diclofenac clusters with the antibiotic ciprofloxacin. When we compare the 2 results directly, we see that there are robust and similar clusters identified by both tandem mass spectrometry and MS1 only acquired data. The dendrograms are visually similar, where major clades are either the same or similar, and an objective cophonetic correlation coefficient of 0.625 demonstrates how similar the results are at both data acquisition levels. In conclusion, MPC can rapidly screen microbiomes and can be used to prioritize treatments of interest for deep metaproteomics, saving time. For example, we can prioritize further exploration of non-antibiotic drugs that cluster with antibiotics. We can use MPC for biomarker discovery by looking at features inside of clusters that demonstrate high correlation with samples or conditions of interest. And this will be especially useful in clinical studies where rapid results are required. But in the end, this is our approach to more efficient, resource conscious, and intentional metaproteomic research. And with that, I'd like to thank you all for listening to my talk. I'd like to give a very large thank you to my two co-supervisors, Dr. Daniel Figues and Dr. Mathieu Lavallee-Adam, as well as all my co-authors on this paper, Dr. Jimin Ning, Dr. Loyan Lee, Dr. Mona Hamada, and Dr. Xu Zhang. Finally, I'd like to thank all of my funding agencies. And if you'd like to read more about MPC, feel free to scan this QR code to be taken to the preprint or visit the shortened URL. Thanks.